All right, let's get started. So welcome everybody. I'm Brigitte Raumann. This is the Research Data Management Strategies for Chorus session. We're going to start the session off with a talk from Aaron Deal from Glencoe Software, who is a sponsor of this meeting.
Thank you so much. Thanks everyone for allowing Glencoe to speak during this session. Very sort of aptly paired session of the sponsor. We do a lot of image data management at Glencoe Software, so really happy to tell you about some of our new tools where we're expanding our ecosystem for image data management using our product, Omero Plus. So to start, I'm going to dive right in because we have 10 minutes here, and so I want to make sure we get right to the point. Omero Plus is our image data management system, and classically when we think about image data management, of course we need to have the image data in hand, the original files that come off the instrument. But arguably, you can put all these files on a file system, and maybe there's some dated folders in there, and you have some semblance of image data management. But the reality is that those image files themselves are not all that useful without some really essential context, so things like experimental metadata. And so in a system like Omero Plus, you aren't just managing the image data itself, but also metadata that describes how that image came to be. And you're also going to do things with that image after it's acquired. So you're going to produce some analytical data, let's say, that also enriches the value of your imaging data sets. And so that can also be managed within a platform like Omero Plus. Now, you also might want to be able to search through those images. And so it's great to have everything nicely tagged and curated, but can you also find it? Let's say that PhD student's images from 5 years ago, are you able to look back and find that this compound has in fact been used with this organism at this concentration and see what the results were from that imaging experiment? And importantly, you might also, especially in the core facility world, you might want to manage the permissions of that data. So you're not just acquiring data in the context of a single lab, but you're acquiring data and helping users acquire data across lots of different labs. And maybe not everyone should have access to everything. And so an image data management system needs to manage the permissions, how you access and what you can do with all of this image data, metadata, et cetera. And so all of this is running on a server somewhere in the context of Omero Plus, and you as the end user are interacting with this system over the internet or maybe some private network within your organization. And so you might be in the core facility acquiring new data and bringing it into the system. Or you might be doing some image processing. And so maybe it's not you, a human, but a Python script, doing some interesting custom image segmentation, and you want to write that data back into Omero Plus, bringing in more metadata, more analytical data into the system. Or you might be exploring your images in any capacity. And so I think increasingly, image data management is still very much about tagging images with appropriate metadata and making it more findable over time. But it's also the complexity and size of image data today means that just interacting with your images, viewing them, annotating them, sharing them with a colleague is a large task. And so you can't just double click on a file and take a look at your images anymore. You may need to be on a particular workstation with the right software installed. It needs to be powerful enough to be able to actually read and load that image data. It needs to be the place where you downloaded the image data to. If you need to go to this other workstation, now you need to download the files there as well. So it makes it really challenging just to interact with image data in the first place. And so I'd like to talk a little bit more about how Omero Plus solves that problem for you. I'd like to compare it to how we interact with TV shows and movies. Classically, it was maybe a fun task, but a larger task to find the movie or the show that you were going to watch on a given night. But now we all know that we have a variety of streaming platforms that we can use in order to explore a massive library of movies and TV shows. And we know that we don't necessarily have all of those files on our phone or on our television or on our laptop. And you also know that you don't have to be on the same device. You can watch season 1 on a laptop, and then you go home and watch season 2 on your television. You know that you can be on any device because you don't have that data locally. You're streaming it from the source, in this case being Netflix. And so can we do, and actually I'll just comment that this is maybe an easier problem for Netflix in the sense that they might have more users than Omero Plus, but we're talking about for a given hour of video, it's a few gigabytes in size. And I think many of you are probably working with imaging data sets that are more like terabytes in size. And so you can think I have a harder problem than Netflix to solve. And so what if we could have the same experience with our imaging data? And so in the bottom, that's a view of Omero Web, a web interface, a browser-based interface where I'm scrolling through a number of images, and I'm opening one up in our viewer called Path Viewer, and one of those images could easily be hundreds of gigabytes in size, and the collection of images could easily be a terabyte or more, and you might have different sizes of your imaging data sets, but they're quite large. And so we're following the same model of streaming our imaging data. And so using Omero Plus, you now are able to interact with your data via the web browser, which means that you don't have to be on a particularly powerful workstation. You don't have to have particular software installed. You can use whatever browser you happen to have installed. And yet you can explore your image data. Here I'm just highlighting viewing, but you could annotate your images, share them with a colleague. You could even kick off an analysis run from the web browser. And so just a little bit more detail on what I mean by streaming the data here. So in the case of Netflix, we're talking about frames of video and audio that you use to watch your videos. And in the case of an image, we're talking about loading individual tiles of our image. And so we don't have to download all the data in order to interact with it. Similar to how you think about interacting with something like Google Earth, as you zoom in and out, you're loading just the data that you need. And so that's how you can interact with a relatively large imaging data set, because at a given moment in time, you're interacting with a very small amount of data, say a single tile of the image, more like a megabyte in size. So just a little bit more detail here in terms of how all these components work together and what the components are. So you've got a bunch of image files, and those are coming off of your microscope. And those are in some file storage. Maybe that's already the case. Maybe you have some massive storage infrastructure where those images are sitting. And by the way, they're in all sorts of different formats because they come off of lots of different microscopes from lots of different vendors. And actually, that can all stay the same. Omero Plus reads image data in its original files from that file storage that you specify. And Omero Plus also has a database. The database doesn't store the images stay where they are. That's kind of the bulk of the sort of data volume that you have. But things like metadata and regions of interest are things that are written into the Omero database. And then you, the end user, again, are interacting with the system over the internet via a browser. And so we have viewers like Path Viewer and other interfaces that are kind of tailor-made for exploring scientific data sets. We also have a lot of opportunities to extend the system. If you want to write a custom script or even a custom web application, all of that is possible with our open API, so we really don't try and create this lockdown system where you can't get data in and out. It's really custom to what your needs are. And so the last thing that I wanted to share is that we're expanding the Omero Plus ecosystem because we want to make this even more achievable for different kinds of organizations to work with image data management and get started as soon as possible. So classically, the way that we do image data management, the way that we deliver Omero Plus as a product is kind of a classic enterprise model. And so it's very customized to each of our customers' requirements, and largely this starts with requirements from the IT side of the organization. We have these servers we want you to use, we have this storage that we want you to use, this is the operating system that would work best for us. And so we have those conversations, and we will continue to do that. It's something that we love to do, and we kind of tailor the architecture of the solution based on what IT really requires from a security or scalability perspective. And that's because we support the software of Omero Plus, but your IT actually hosts the software. So it's not a SaaS solution, it's running on premises or in the cloud, but your cloud environment, your data center. And so IT gets to sort of set the rules of how that's deployed. And increasingly, we're seeing more and more customers who want to work in the cloud. So we wanted to understand, could we offer solutions in the cloud that made it easier for different kinds of organizations to work with Omero Plus? And so the first is Omero Plus Essentials. This is more of a self-service solution for Omero Plus. So you support and host the software. And it's more of a pay by the hour, consistent with some other cloud charges, so that you can get started, run Omero Plus for a day or a week, or for a year if you so desire, and the cost structure is basically nothing up front and only payment by the hour for the Omero Plus sort of license component, and of course all the infrastructure costs in the cloud. And on the other side, we have Omero Plus hosted, where Glencoe software actually does everything. We not only support the software, but we also support the infrastructure on which Omero Plus is running. And so this offsets that IT burden because effectively Glencoe software becomes the IT part of your organization that manages Omero Plus. And so these are kind of bookends for our classic enterprise deployments. And that's me at times. So we have these feature comparison tables. Won't go into all the details here, but very happy to discuss them in more detail. We have some little handouts if you'd like to take one of those with you. We're still around for the rest of the day at booth 228. With that, I'll just say thanks so much for listening. Okay, yeah, if there's any questions, happy to take any now, or we can chat after. Okay, great, thank you so much. Thanks for having me.
Thank you, Erin. And thank you to Glencoe Software for sponsoring. We appreciate it. Where is the cursor? Okay. Problem number 1. I don't see the cursor. Oh, that's because we're on 2 screens. Got it. There it is. Okay. Thank you. And sorry, I'm not a Windows user. Where is the slot? Oh, there it is. Is that it? Yeah. Okay. Great. Here we go. And okay. All right. So again, welcome everybody. I'm Brigitte Raumann, and I am from the University of Chicago. And thank you very much for coming here at the very end of the meeting. I appreciate that. I'm going to give you a really quick intro to myself and then an intro to the topic, and then we can dive quickly into questions. So as I said, I'm at the University of Chicago. I am with the Globus team. Globus is a software tool that's designed for research data management, and mostly those management tasks that are more mundane. So kind of the more boring tasks that any science discipline needs to do. So there I manage the product team, the user support team, and the compliance team. So just really quickly, as I said, Globus is really part of cyber infrastructure, so kind of the lower level tasks that any kind of data intensive science needs to be efficient at. And so it's used across domains. It's not specific to any one domain, so it's used by high energy physicists, cosmologists, climate scientists, scholars in the digital humanities, and of course biologists. It's used widely across the globe, across scientific disciplines, as I said, and across most of the larger research institutions in the US. Just really quickly, I'm going to touch on a couple things that Globus can do. So first of all, if you've heard of Globus at all, and I wouldn't be surprised if you haven't, unless you're a storage system admin, you know Globus is a file transfer service, so that's probably what you've heard of Globus, and it certainly is that. So it's been around for decades and has been managed file transfer for the nonprofit research community for many, many years. And many cores use it just for that, so to move data off of the instrument quickly and efficiently to storage space, to archival space, to space near compute. A lot of cores also use it to share data. So there's a data sharing feature, and many cores use it to distribute the data to their users, whether those users are internal or external. We also have services for remote function execution, services for searching, ingesting metadata, and then searching your data sets, and also for automation. And by automation, I don't mean necessarily automation within one system. It's really task orchestration. So Globus is especially good at managing tasks across different systems at different institutions, for example, in different storage systems, different compute centers, and orchestrating all of those dependencies. And everything I've talked about is available through an API, and we use our own APIs to provide clients a web client and a command line interface. So if any of this sounds interesting, come talk to me after this session. I know it's very abstract here. The best conversations really are usually when someone talks to me about what their problems are in their core, and it's possible that Globus can help. That's usually the best way to start a conversation. Okay, but Globus isn't the topic today. The topic is research data management, and that's really far broader than any one tool or any one solution. So, and it's an important topic. I've heard it come up in multiple contexts during this meeting in many different sessions, and clearly, given the attendance at the end of the meeting, right, it's top of mind to many cores. And that's because for many cores, research data is what they're producing, it is their product, it's why they're there, it's critical to their success, especially genomics cores I'm thinking of, or imaging cores. So it's very clear why that might be important to those types of cores, but it's also critical, right, to cores where data isn't necessarily the primary product. Maybe it's producing antibodies, maybe it's producing transgenic mice, but the metadata associated with that product is critical for the value of what that core is providing. So we have 3 panelists today, and they each have their own unique perspective on data management. We have April Williams from the Salk Institute, who is the director of the Integrated Genomics core, Integrated Genomics and Bioinformatics core, at the Salk. And Stuart Levine, who is the director of the Biomicro Center at MIT, and Scott Edmiston from Stanford, who is the director of Research Data Governance and Privacy. So we got together a few weeks ago and decided what we wanted to do with this session is have, rather than 3 sort of longer independent presentations, we're going to have 3 very short presentations just so the speakers can introduce themselves, so you know kind of where they're coming from, and really reserve the bulk of the session for questions and answers. We want to hear from you. We want to also hear opinions and shared experiences, and so I hope we can get a lot of audience engagement. So who wants to go first? Stuart, you're closer to me, so why don't you go first?
I go first.
Okay.
Okay. Okay. Hello, everybody. Again, thank you for joining us and not already being at the pool. I'm Stuart. Most of you probably know me, but I am the director of the Biomicro Center at MIT. The Biomicro Center is an integrated genomics and bioinformatics core facility as well. We really work with folks everywhere from experimental design, library preparation, short long-read sequencing, data analysis. And in the past about 5 years, we focused a lot on data management. I'm just going to begin very quickly. What do I mean by data management? I want to put some definitions out there. Other people on the panel may have different definitions, but this is what I use. There are kind of 3 levels that I think about this on. First is data storage. There's a lot of challenges associated with data storage. These are questions like what physical storage is, networking, data integrity, quality assurance, data security levels so that you're not stealing somebody's social security numbers. Then you've got questions of data lifecycle. Those are separate. Those are questions of governance, who owns it, the different data, how is it backed and provisioned, in terms of being on hot storage versus glacier, as well as legal types of questions of how long do we need to retain it. And then lastly, questions about data management. And from our point of view, this is questions about really starting to get into questions like fair data principles, making data findable, accessible, interoperable, and reusable. Rigor and reproducibility, questions that ABRF has focused on for years. Repositories, ontologies, data tagging, metadata, and data provenance. And each of these is important. They're nested, right? You can't even think about data management without thinking about lifecycle, which you can't think about without thinking about storage. So as part of our data management efforts, we've been asked to serve as the data management team for a large number of multi-center projects. And that's kind of where this began. So these are projects from the NCI, including the Metastasis Research Network, as well as the CSBC Consortium, MIT's Superfund Research Program, so where they're looking at metastatic cells or systems biology and GBM. Here we're looking at contaminated wastewater in sites north of Boston that have led to high levels of cancer in the towns, but we've got devices put in the Mystic Lakes we have to record data from. And lastly, Impact TB, which is from NIAID, which is a 3-continent, 8-center, 3 patient arms, 2 monkey arms study of tuberculosis and how to create infectious vaccines for it. And we needed one system to rule them all, because I wasn't doing it 4 times. So we found a system called CIPP, and that's fundamentally what we've used. We've built a wraparound, but the core of CIPP is it's incredibly reductionist. It has this idea that your samples are nodes, and they're described by tables. And your experiments are edges between those nodes and making a graph, and they're described by protocols, which are Word docs effectively. And from this, we can build networks. And so CIPP is software that's used more in Europe than stateside. It also has an advantage that it has its own repository called FairDom Hub. And this is a public instance of CIPP. It accepts deposition of CIPP records. And it allows basically anybody to deposit anything. This is a very abbreviated talk of what I normally do on this, but this is an example of the data from our Impact TB program. We've about 65,000 samples in the system at this point. We have an internal version with 65K. There are about 10,000 of those that are in the public domain. Most are internal. These are of every type of flavor. This includes images stored on a marrow, as well as flow cytometry data on import, sequencing data on SRA, dbGaP, Geo, and where we have no other repository, we'll put it on Zenodo. And so we're connecting to data wherever it is. Just quickly, again, we are a data management core. So there are some what does it mean to be a data management core? There are some advantages, economies of scale. It's the same tool we're using for all of these platforms. The skills are not critical as a future faculty member. As a bioinformatics core, this is a challenge for us normally, because if we're doing somebody's analysis for them, that makes them a less good candidate to be for their next job. But that's not true for data management. We can do it for them, and it's okay. It's okay to have some bread and butter here. We're not reinventing the wheel every time. We understand the resources. We paid attention to the repositories. We know the metadata they need. We can get that information up front. And as core facilities, we generate the data anyway. There are disadvantages. You still need new people. As many of us have found, you need data managers on site to gather that data in. We don't know with specific field ontologies ourselves. A lot of people in those ontologies can look down on us because we don't know every type of PFAS out there. Enforcement, we don't have sticks, we don't have carrots, we have twigs, we can annoy people. And we don't, what's the institutional support behind this long term, right? How are we going to pay for the data? This doesn't solve that problem. It simply solves a way to orchestrate everything. These are the important people. These are the people on the team who are doing our data management. We have 2 data specialists, a senior and junior, who do a lot of the training, some of the coding, and a lot of data management, IT specialists, and a bioinformatician in database systems management. All the key people who help develop the tool and all of the programs we're working on. And thank you to all the researchers who gave us the data. So I'll pass it on.
So hi, I'm April Williams, as Brigitte so nicely introduced. I'm the director of the Integrative Genomics Core and Bioinformatics. I don't know what it's half one name, half the other, but at Salk, I run a team of bioinformatics scientists, so there's 3 bioinformatics analysts, and I also run the short and long read sequencing core. And I'm also the sole data manager and data curator for all of the cores, not just genomics. But we've made the most progress in genomic data management, but we're considering now, how do we roll in other cores? And so this is really a story of like what one person can actually do. If you don't have a team like at MIT or other big places, you can still make a difference. And as Stuart said, the goal is fair data, but sometimes, perfect gets in the way of good. So if you can at least tackle a few of these, then that's better than nothing, and that's kind of been our philosophy. We're trying to get everything to be fair, but that's not always possible from the get-go, and to build systems that or at least have these principles in mind is the goal. So how is our data management set up? So we have the cores at Salk, where we're generating sequencing data, microscopy data, flow data, mass spec data, all kinds of other data. And then we have a bunch of different storage systems, DDN, Ceph, VAST. We have a couple petabytes of storage. We have about 1 petabyte of genomic sequencing data that we've acquired over the last 20 or so years that we manage for everybody. And then we have compute on-prem, and then we're moving towards more cloud-based solutions for data sharing, and also for the piece that Stuart alluded to, which is how did this get funded? Who pays for this? So that we can maybe move some of the institutional commitment to more direct costs and make the data more findable and usable by the researchers that actually generated it. So for that, we're doing archival storage on tape on premises, but also on Amazon Glacier Deep Archive. We're starting to use a tool called Zero that was developed at the Fred Hutch to do data management, but also empower users to run their own bioinformatics analysis, as we have limited resources, and to give them reproducible pipelines that they can run at the push of a button instead of having to learn command line and programming languages. And then also using bioturing, which is similar, but only for single cell and spatial data. And so one of the main problems is, for all the data that we've generated, it's stored, especially for the genomics data, it's stored, and the metadata is searchable. I can find almost anything. If you come to me and say, my apocryphal story is Gerald, who's been a postdoc at Salk, or a staff scientist now, I guess, for 20 years, said, I sequenced a blue whale genome in 2016. Can you find it? I need it now to submit it somewhere. And I can find it. I can find it in less than an hour, send it to them, look through with the institutional knowledge that, in 2016, Gerald was in Inder Verma's lab. Look through all of those, scrape the metadata from this FASTQ screen to find blue whale, and then send him that data that he generated. But that all only exists in my brain. And part of the effort that we've had in the last couple of years is we have a Starfish license and deployment on campus. So to try to move that knowledge out of just my brain into Starfish. And so if you're not familiar with Starfish, Starfish is basically like an indexing service for any sort of storage. So it can index anything, HPC storage, enterprise level NAS, tape archives, object storage, and even object deep archive storage. And it creates a database, basically, of all your data. That's much faster to search and query and generate reports than the entire data system. And so we can do data acquisition workflows. What we've been mostly using it for is data movement, data analytics, and data tagging with metadata. And so some of that is, I have my file structure of a run that comes off of our sequencing machine. It's very small, so you don't feel compelled to read it. But I know where all of the different metadata that I want to store is stored here. And using Starfish, we can scrape all of this, add that data, associate that metadata with any FASTQ file that we generate. We can generate reports on the storage, which allows us to see, if somebody's directory is getting a little bit out of hand, we can go through and clean up and look to see where the major major things clogging up our storage are, and we can build reports in Redash based on the Starfish database. And so those reports, my boss may ask me like, how many sequencing runs, how much data are we using? And but I could always generate those reports, but now I don't have to do that manually. I can use the Redash dashboard to generate those reports. And so we also use, like I said, I'm not a vendor for any of these tools, so I can tell you about how we're deploying them, but I can't tell you, that much about them. But we're using Xero now to also expose workflows and move this into the cloud. So we have, a listable, you have all the data sets associated with a given project and the cost associated with that. And we can now have pipelines and and more metadata associated with that the user can query and search in a web browser context without having to come back to me to get their data. And so that it's always findable for them for hopefully, as we build this project out, all the data that they generate as part of the genomics core and other cores on campus at Salk. So yeah, I don't have an end slide. It's just the start of the discussion.
Hello, everyone. I'm going to break with convention because I'm not going to have slides. I'd actually like to be a little bit more Socratic and talk a little bit about my role and some of the things that we're doing at Stanford with regard to data management. Scott Edmiston, I'm the Director of Research, Data Governance, and Privacy at Stanford. We have a large portfolio of research, almost $2 billion of research, sponsored research, and 6,000 researchers, 50 to 70 cores or service centers, depending on how you define them. My role is a new one at Stanford, created about 4 years ago with the recognition that data and data management is really at the core of the research enterprise, and computational tools and advanced computational methods will define the research ecosystem for the foreseeable future. And so I just want to share some of our experiences and our philosophy around data management and some of the strategies we've taken. First of all, I think data management doesn't have one definition. It has so many definitions. And I like to think of data management in terms of both strategy and tactics. And when I talk about strategies, data management has a goal. So the overarching goal, as we're all familiar with, is data management supports rigorous and reproducible science to advance fundamental biomedical discovery. And that's the overall concern. And that actually can be the means of other goals of data management, because data management can also serve the goal of compliance with sponsor obligations. And a prominent one is the NIH data sharing and management policy. And so if you sort of use that as, even though there's a lot of research that isn't NIH sponsored, that at Stanford, it really becomes almost like the template, the lodestar of our data management strategy and compliance agenda. And it's been really useful because NIH has really taken the lead in terms of some of the open science principles, FAIR principles, and kinds of tools that can help researchers at a very basic level to prepare and curate their data for repositories. Now the dark side, not the dark side, but the downside, of course, is that there are unfunded mandates aspects to this. I mean, when everything looks good on paper in terms of data management and lifecycle philosophies around data, but when you come down to it, people will say, well, some of these interpretation issues about what data do you store, those kinds of issues are very complex, because NIH might say one thing, but maybe this project has other data contributors where different terms and conditions of DUAs or governance that comes from human subjects protections, there might be privacy and security issues, there might be state laws. So when this whole parallel ecosystem of data management frameworks, security frameworks come together, it can actually paralyze a project. It can paralyze the people who are responsible for research data management. I'm grateful for tools like, as was just described, that can manage some of these pieces, and we're grateful, and we're especially grateful for really vital communities that create community standards around metadata like Omero and some of those sort of level 5 sophisticated frameworks. But there are a lot of disciplines that don't have that kind of sophisticated community that emphasize metadata standards and sharing and life cycle tracking and provenance of data. And for those situations, we really need to navigate for them. We have to provide solutions when there are gray areas, when these laws that were created and didn't have things like LLMs and AI and machine learning in mind. What do you do in that vacuum? And usually the answer is to try to come up with very practical solutions that are based on, again, the integrity of the science. The interests that inform the law in the first place, even if some of the technical pieces are difficult to follow. So I'm going to share a practical example, not because I'm authoritative. This is my first conference. I'm here more to listen than to talk, but I hope it stimulates some other blood, sweat, and tears stories from you in the audience, and we can share together. So here's one example. Google gave us free storage. And what happens when you get free storage? You use it. And that's certainly what happened to Stanford and our other peers around the country in the academic medical center world. And one day, the rug was pulled out. They said, I think we should charge money for that, a lot of money. And it happened to be money that people didn't have. So it created a conundrum. And another, a parallel problem in the area of storage is that, sometimes people leave the institution. And they kind of want to bring their data with them. And sometimes that data can reach into the terabytes and above. And we have a policy within the Vice Provost and Dean of Research that has retention schedules and principles where you're allowed to take data copies to your new institution. Originals are supposed to stay here, but if there's sensitive data like protected health information or so forth, you're supposed to have by policy, a data use agreement between Stanford and the new institution. But in practice, it's like my garage at home. Like when I moved from Boston to the Bay Area, I didn't really put metadata tags on the stuff in my garage. It was just, everything was just thrown in there. And so when people leave, it's very difficult to disentangle things it's a data management problem, fundamentally. And so, what am I trying to say when I'm going to this? So for the Google issue, we came up with some solutions. One was this new project that actually leverages Globus. It's called Elm Storage, which has an automated pipeline to large storage on tape that's run by research computing. I mean, that's a solution. It's by no means a panacea, and we're still doing some beta testing and early adopters. That's one solution. I would recommend in terms of best practices to really cultivate those relationships with UIT and other sources that can help compress the data and provide other solutions. Often the cloud cold storage is often too expensive, so we're looking at different kinds of on-prem solutions, co-location, and other kinds of things like that. So that's the kind of creative thinking that we try to encourage. And then, so my role, I'm often called in when, there are these gray areas in terms of when compliance is a must-have versus a nice-to-have. So it really becomes a team sport, and we really try to, maximize data really across the life cycle. So I think. So we'll start there, and then hopefully we can generate some audience participation. Thanks.
Okay. Yep. All right. Oh, sure, Chris. Go for it. No.
I'm curious, there's a lot of people in the room. Just a show of hands, how many people think they benefit from good data management? Okay, wow. Not quite as many as I thought. But let me ask you a question. I mean, you're talking about like you don't put metadata tags in everything you throw into your garage. I get that. The problem is I live in your garage, and so I have to do this. I benefit from this. How does the institution benefit from good data management practices?
Great question, and there's always a gap between the ideal and the reality in a situation. I think each one of us is a data steward, that we're responsible for data somewhere along the life cycle. And so how does the institution benefit? Just on so many parameters. For compliance, you reduce risk. You enhance institutional reputation by having solid data management. We're creating gold standard data sets, because if you don't have a gold standard or regulatory grade data and you want to create an FDA regulated product, they're going to say, no, sorry. Or you want to be a trusted partner in these data sharing networks. If you have a multi-site study, if you have a collaboration, they want to know if you have good, sound data practices along parameters of data security, privacy, compliance, corresponding to professional standards, domain-specific standards. There might be a generalist repository. There might be a domain-specific repository. There's so many, there might be professional societies. So there's not one source that rules them all, but institutions have this ultimate data stewardship responsibility, and it applies to all of these things.
So it's a good point. I think, however, we leave a piece of this equation out, and it's a really important one, and that is the reward system. Chris, it's a great question. How does the Institute benefit? The Institute always benefits. How does the researcher who generated that data benefit? And the short answer is they don't. Right? These standards, show of hands, how many people here are informaticists? Okay, you're the problem. You got in the ear of the NIH and said, I'm sick and tired of having to get these papers and figure out like what they actually did. And so all those wet lab people, like the NIH is going to get a lot more work done from us and better ROI from us if the data is actually well annotated. And so now to submit a paper, it is so much harder if you're a wet lab scientist. So when I was a biochemist, it was easy. I don't envy the kids these days. The amount of things they have to write down. So, I mean, I say that being an informaticist, let's be clear. I'm an informaticist as well. But it's tough for them, right? It's tough for them to do this, and they're not getting a reward. And so it gets this, again, what's the benefit? There have been all sorts of things proposed about like DOI numbers if you generate a good data set. I haven't heard of a single person getting a faculty position at a top institution based on that, though. Right? It's not there. And so it's a really tough, it's your problem. It's in our head. It's in your head. It's in my head. But how do we get it down and simplify that process? So I'll leave it there.
But Stuart, do you agree that it's important to actually have that written down, annotated? So are we coming from that position to start with?
Yes and no. I'll say yes and no. I think, yes, it's absolutely good to be doing reusable and reproducible science. It's good to write it down. However, the standard that the NIH has that every piece of data shall be made public, and that will improve reuse and reusability. I think that's still a premise. I don't know that I believe that that is proven. It's one thing for a large study, right, where you're doing something that's going to be put in Cell as a resource, or like these large ENCODE or something like that. But for somebody's individual R01 and somebody's Western blot for that R01, or somebody's flow cytometry data, are there any flow cytometrists here? Your repositories are awful. And we yes, they're horrible. But the question is anybody going to ever look at it? right, to reanalyze somebody's flow data. I mean, maybe, but the amount of effort we have to do on my team to deposit flow data into import or into flowcytometry.org, it's awful, right? And so I'm not sure that it's true.
So I have some good news and bad news on that. So first of all, Stuart is a giant in the field and has so much experience that I respect. I think everything he said is true, but I think you don't have to save every shred of data. I don't think the NIH policy really requires that. It's only the data that's necessary to validate the results. So a lot of researchers come to me and say, hey, Scott, we have to save every single tittle and piece of raw data, and that's going to be terabytes. I said, no, okay, calm down. You can actually just, you can even save metadata potentially. You have to consult your discipline and everything, but just enough data to validate the results, and that can actually be a fraction of what the total amount of data is.
Is that to validate the published results, or which results
The
are you
final
talking
results,
about?
whatever the final results are, I mean, and that's the case.
Okay, Stuart, I'm going to push back a little bit, okay? Because I love doing that to you. So what about the concept of if more of that data was findable, if it was searchable? As research funding will shrink, as we all kind of are starting to feel that that might be a possibility, will this be, having good data management and good annotation standards allow us to get more research information, do more discovery with experimental data that's already been generated, if we can access it? Is that the reward that we have to look towards as a scientific community when we didn't have to worry about it, when we had all the space in the world, we had all the money in the world to be able to, the biggest garages ever, to not worry about having it annotated? That's where I think we may run into the need for this. What do you think?
Again, look, there's a reward for the graduate student themselves who's left 5 years ago. That's the tough part. So what we've done, the way I think about this with our stuff is, I think data harmonization and data provenance are the enemies of each other. That there's a real clash between harmonization and provenance. And one of the real challenges, I think, for researchers is that harmonization effort. That, if anybody's been involved in a big project, and it takes a year before everybody agrees on what we're going to call everything, that's not, I understand why you're doing it, but boy, that's not the fun part of science in any way, shape, or form. And it makes it hard and it's constantly coming down to the researcher. What we've thought about is, instead, our system with CIPP, because it's unarchitected, it doesn't have a mouse where these are the fields that thou shalt put in. We're working with the researchers to gather whatever they can. We try to think about it from the solution we're trying to provide them is to be greedy about metadata. Just suck it all in. Everything you have. Don't worry about what you call it. Don't worry about anything. Just get it in now. Get it in before that graduate student graduates, and now you have to try to go through the damn notebook 3 years down the road and figure out what the hell they did. And LLMs can probably do the harmonization, right? They can read text, they can look at images, they can add these words, and whether it's F or female or whatever, they can match those. So we can figure out the LLMs faster than we can figure out the time machines to get the provenance. Because once provenance is gone, it's just gone. And so that's been our approach to it. But the reward is, how do you get that graduate student to do it? Our thought is lower the energy, make it as easy as possible. But that's all I can think of.
Yeah, I kind of to take it down a level, because we're talking about, if anybody's ever tried to analyze data from even SRA or GEO, and you pull it down, and what people deposited isn't really what you think it was, and you have to redo it all anyway, and so what was the effort of putting it up there? I think one of the ways that we can add value is not just in thinking of depositing it into public repositories, but to creating robust lab repositories within a lab or within an institute where the lab can find the data. So if that flow experiment has already been done and generated that results, and the PI remembers it a certain way, but that guy's gone, it makes it worthwhile for the students and the researchers when they don't have to redo work and re-spend money that they've already spent generating a data set. And that's where I think some of the value will come in. The value proposition will come in to individuals within a lab.
What do you think the role of the core is in collecting, or even responsibility, if any?
That's
does
going
that
to be
rest?
my question.
Excuse me. Oh. Well, go ahead.
Thank you. I'm an electron microscopy co-director. We are a user facility. People use microscopes. They save the data on a local hard drive. I guess my question is, what responsibilities do I have or my core have for that data? Do I have a responsibility? And if so, the question is, for how long? If I don't have any responsibility for it, the question is, for how long?
I mean, so I run a genomics core, right? So we have taken the position that the responsibility is ours forever, which may change. It may have to change. But for now, I have every single sequencing run that we've ever done at the Institute saved. It's backed up on our, obsolete storage drives on-prem, on tape on-prem, and then in the cloud. So there's a copy of it somewhere if we need to get it back. But that's much easier said than done. In cores where users are generating the data. I generate all the data, so I have much better control over how to collect the metadata, where it is. I totally understand where to find everything, but in user-defined cores, that hasn't been our policy either. The microscopy core is dealing with that now, where everything is on user drives, and we're trying to come up with a more concerted solution to managing all of that data, and then also just enabling the researcher to be more responsible with it by providing them with systems where the data gets sucked in, and also, now in today's funding climate where they can be responsible for paying for it as well, because the cores don't have the funds to pay for all of that on-prem storage.
If I could just add one more thing, which is that I've already had visits from our export control people asking how our users are going to store the data if they have those controlled projects. And I think at its simplest, it's plug in an external hard drive, save it to that, unplug it, and leave. So that's our export control strategy right now.
That's another topic altogether. But I want to address the responsibility piece, and I would say it varies among cores, but I think ideally the core manager has an interim stewardship responsibility while he or she has possession of the data, but that ultimate responsibility is the PI. And I think good policy makes that role and responsibility clear up front. So we've had problems where researchers dump data and forget about it, and then data becomes this ghost data and people don't track it, and it's not a good situation. So that's bad data management. Good data management is where roles and responsibilities are clear and follow through on.
Hence me asking for how long.
So it's interesting, like some in the old days would have it for 10 years, some would be 3. And the Vice President and Dean of Research, we have a floor of 3 years. And then we purposefully leave it open because there might be terms and conditions for other projects on a project-specific basis. That's frustrating to people because some people just want a hard and fast answer. But if we gave a hard and fast answer, that might actually conflict with some of our legal obligations. So it's a case-by-case basis, and hopefully you have a resource that can sort that out for you.
I wish.
Yeah. So I think the actual answer is legally none. Right? It's the lab.
Yeah.
Because we can't know those terms. Like, it's not my job to know the legal responsibility.
Great point.
I will say for images, I would talk to Amera as a place to put it, certainly.
Yeah.
Yeah, just on that note of how long, as an NSF major facility that I work for at the Mag Lab, they like to use phrases like long-term without actually attaching any numbers to that phrase. And when you ask NSF program officers, who are great people, I'm not criticizing them, they don't really have solid or consistent answers for you. So it can be a little bit frustrating.
So I have actually a lot to say about that conundrum and some other related issues, because as part of the NIH policy, and other sponsors, even private sponsors, want the data to be held indefinitely to maximize secondary uses, but they're not necessarily paying money for it. And so, if the money can only be charged within the span of the project, what do you do for that tail end? So there's actually an interim solution, which I'm sharing with you today, but I can't honestly say that we've rolled it out perfectly as Stanford by any means. But you can charge a reasonable cost-based fee to make the data available asynchronously, in the sense that you don't necessarily have to put all the data in a publicly available repository where you press a button and you automatically get the data. You can say, well, it is available, but you do have to take these steps, and it can lessen the administrative burden and cost for an institution. I mean, that's not going to answer all your questions or solve all your problems, but I think those are the kinds of creative solutions that we have to deal with.
Right, and to get to my full question, or maybe it's more of a comment, but thanks for your cautionary tale about Google kind of offering this free storage and then trying to come back later and then send you a bill for it. There was a session at last year's NSF Research Infrastructure Office meeting where we talked extensively about the benefits and drawbacks of moving data storage to the cloud for the long term. Some facilities were moving to the cloud for cost and convenience reasons, and some were moving away for cost and convenience reasons. So it's kind of interesting the different perspectives facilities have, which sometimes depends on how much data they're generating. But there were a lot of concerns about data ingress and egress costs, long-term storage costs, and migration costs, because if those become too high, then you effectively become locked into a platform.
Sure. I mean, I don't think anyone believes that there's going to be a one-size-fits-all solution. I mean, there's going to be cloud solutions that are absolutely great, and then there are going to be solutions that are cold storage solutions that might be, tape might be fine. There might be some on-prem storage solutions and everything in between.
Thank you.
Yeah, hello. So I just have a question about where we work. I mean, it sounds to me like talking about storage of images or, I don't know, alignments or something, but what about the analysis? I'm just talking about in my core, like part of it is their analysis, could be simple or more complicated, and it might relate to the data in various ways, to some or all the data or various types of data. How do you deal with that? Thank you.
Sure. I mean, so it hasn't been a super elegant solution so far. I mean, we have workflows that hopefully are reproducible, and we save, like a snapshot of the environment or whatever environment we loaded. And those are the things that are most saved, and we can get rid of most of the intermediate files as long as we feel that we have a way to get back to, to regenerate them if needed. And that's kind of where some of these more, well, they can be cloud or on-prem based solutions like Zero or Galaxy come in, where there's a reproducible run workflow that you can get rid of all the intermediate files and keep but there's the data provenance is saved, and the workflow can be re-executed with the same environment again.
So, but I mean, for example, that whale project that you did in 2016, the analysis, like, is it somewhere in there?
That was for, that was just getting the FASTQ files into Geo, because he had to submit it for his, otherwise he's not allowed to submit it, his analysis about it. We don't do analysis for everyone. We're not like the be-all, end-all. There's bioinformaticians in each lab, and so then that is a separate problem of how well are they tracking their usage. And that's why we're trying, pushing really hard to move into a more concerted platform so that things are more reproducible and more able to be done by non-experts as well, like you were talking about in the Nextflow session. Like, instead of even having to run Nextflow now, if in some of the, in some of these environments, you can just click the Nextflow pipeline and type in the parameters in a GUI, and then it executes.
Right, because I mean, it's a situation, you have the data 1 year, and then 3 years later, you decide to move everything to the cloud, and then the analysis is somewhere in there, and then now the data is who knows where, and, how do you track all that? That's what I'm wondering, tracking.
So for tracking, I mean, we use Starfish now, so that has the data provenance tracking. So it has, since, it's a database, there are snapshots, so I can see what it looked like, 1 year ago, 2 years ago, and then also tracks all transactions. So if something is moved, copied to, GDA, it's recorded. If something's moved permanently to GDA, it's recorded, and we can somewhat go back. But yeah, it's not a perfect system where they're linked forever, which is, also a problem. So sorry, I don't have a good answer.
A lot of these projects are saved at a lot of this data that you're talking about is saved at the project level. I mean, like for instance, a lot of data scientists will save it on their Jupyter notebooks and these kinds of pieces to reproduce things. But it definitely varies by domain.
So within Seek, the way we handle this is just like I mentioned, like samples as mice and so on. All of the files are also samples, right? So we're pointing to those files, whether they're raw files or analyzed files or models. We have quick naming conventions for them. And the edges are your Jupyter notebook or your Nextflow. And so we're recording, at least we have the ability to record, whether they're putting in the data or not is the challenge, exactly what they did, exactly the workflows. The method can point to your Git repo, okay? And so we can go as far downstream as the user wants with those files where they are, and if those files then are moved to public repositories, we simply change the pointer on where that file is now kept.
So I'm glad Stuart mentioned Git repo, because that leads me to a practical tip, which is that we all should be leveraging existing paradigms for data management within disciplines like computer science, software, and engineering, like secure software development lifecycles and DevOps that can, automatically provide some of these data management pieces without us reinventing the wheel.
So back to roles and responsibilities. So someone said, I think it was Scott, you said good data management is defining the roles and responsibilities. And so as a core, any advice for how to get those defined, right? So where would a core director go? What would you do? Does your core have them defined? Is it in a data management plan? Is that the solution? How many people are using data management plans, and is that a good way? Is that what you're talking about in terms of defining who's going to do what?
So being a lawyer, I'm going to say it depends. I think for any given core, and it's hard to apply this to every single core, but I would say that every core has a core set of roles and responsibilities. And to the extent that they're defined, trained, and communicated to researchers and end users varies. And I think the approach that we want at Stanford is to create as many self-service tools and so forth to do that, but maybe even to have dedicated working groups that are interested in these issues. I'm just talking off the top of my head. I'm not sure that these things actually exist, but we don't even have a data management core like Stuart has. I think that I'm actually taking notes like right now, like going back to Stanford. Maybe we can talk to him later, but it's such an imperative. And I think that if the core responsibilities are data focused and not instrument focused, we're going to get farther. Because I think most of people's heads are around the instrument and its maintenance and so forth, and not so much on the data. And we want that paradigm shift.
Yeah, I actually have exactly that question, right? As a community, what do people feel is the responsibility of cores? And is there something that we can do as a community to develop those roles, responsibilities? April is one of our cores at Salk Institute who does have a good structure for data management, and she can find data from 100 years ago, right? I have another flow cytometry core who they feel the responsibility is to generate the data, hand the data off, and they're done with it, right? Having two competing, or two non-competing but different practices for the same set of investigators in an institution that has only 50 faculty members, for me, creates a real problem. Because there's a different set of expectations, and the community that we're responsible for, April and I, have different approaches to data management. What I would love to see is how can this community at ABRF help set some standards with regard to roles and responsibilities. Is it impossible?
Can I ask you a clarifying question?
Sure.
Do you work the cost of what it costs you to store the data indefinitely into your recharge rates? Do you charge the users for those things? And is that something you think about? We work in the cost for storing it for at least 1 year on our hottest storage level. That is included in the price of genomic sequencing for us. And there's also a charge included for data curation to pay for someone's time to know where it is. But again, it doesn't cover it indefinitely. So that's kind of how but there are different expectations for different cores. And I think one of the reasons genomics has taken this, I save everything, is because unlike flow cytometry, if they can't produce those FASTQ files, they can't publish the paper with that data, and it's totally lost. And they've lost the opportunity to publish it and let the world know about their discovery, but the NIH or the funder has also lost the money for that experiment, and it would have to be redone. Whereas, if you can't produce the original flow files, I don't think anybody's gonna beat down your door. But now they're required to have SRA numbers to submit, to even hit before they hit the submit number, it's the submit button. So that's kind of where that's why it is that way for genomics and not other things, I feel. If if the funding agencies or journals took the step to say that you need to, put your FCS file somewhere,
Mm-hmm.
then we'd have to change how we operate.
So I think also at Salk, though, we have this mixed model where we have a new CIO who we are working with him to try to define institutional responsibility. So I kind of look to you, Scott, in terms of, how at Stanford, I think it's the institution has to define also what they believe their responsibility is. And if it's only, if it all comes down to the PI making those independent decisions, you've got, 400
Right, yeah.
independent approaches, which
Absolutely.
isn't economical, nor efficient,
Absolutely.
nor compliant sometimes.
Yeah, it can't be a situation where 100% of the responsibility is on the PI. That's true. And then conversely, it can't be a situation where 100% of the responsibility is on the institution. I mean, it has to be shared. And you asked the right question, which is how do you divide those roles and responsibilities? I'll just tell you again, at Stanford, the cores have a reporting line into the Vice Provost and Dean of Research, where I'm based, and it's the central administrative unit for research. But again, I don't operate by an oversight command and control model. It's really a trusted advisor. I and my colleagues look at each project, each core, its mission, and the data that we have a responsibility for, and see which services. I like to use the term service-oriented architecture, like borrowing from computer science, to think about, okay, what institutional support services can promote this culture of proactive, prospective planning for data across the life cycle. And that's our goal, to create a world-class data infrastructure that powers innovative breakthroughs and fundamental research. So, I mean, for storage, there's both resources at the core level, but then there's institutional pieces, like I just mentioned, this ELM project at Research Computing, which is going to be a free service. I mean, the institution is investing money that's available for researchers. My salary is being paid by the institution, so I save hundreds of thousands of dollars of legal fees, and you know, instead of paying Stroh's Friedberg to do a big service, I think they save money that way for researchers and things like that. So I could give you like 100 topics where we could say, you know, this is the researcher's responsibility, this is the institution's, and then slice and dice it from different parameters. What I know though is that the vice provost and dean of research does not want to make one single data management manual that applies to all of the cores. Like we are a decentralized organization, and we actually think that it works better in that way. So from the centralization point, we do have a research policy handbook that provides certain rules, and you know, like I said in the retention schedule, it provides a floor of standards that meets most, like, you know, the 80-20 rule. But a lot of the practical decisions are going to be made at the departmental level, at the PI level.
Going along with the community aspects that we've been talking about, are there other domains that we should be exploring for inspiration? It feels like here at ABRF, data management is maybe peripheral to the core focus of what's being talked about. Are you aware of other contexts that we should be exploring and drawing inspiration from? Or maybe not, I don't. No one else is coming.
No one else is coming. It's true. No one is coming to save you. It's just me. It's behind every door. It's just me, all the way down. But I mean, there are conferences, like there are groups of people that are more concerned with this. Like BioIT is a conference where the IT professionals and the biologists get together to talk about, you know, why they're generating so much data, and you know, why they keep buying more things and putting them in the data center, and what we're going to do. So there, you know, and there are vendors that are specific to that. Starfish is obviously not specific to the life sciences, but it's been very focused on the academic world in terms of, you know, easing some of these data management problems across different storage classes and things that make things particularly difficult when you're in an environment where you don't just have millions and billions of dollars to buy huge, one size, you know, totally harmonized data infrastructure, because like I said, we have like NetApps, Ceph, Synology, VAST, DDN. We had any type of storage device that they sold at one point. If it was ever the cheapest one, there was one in the data center, you know, at least. And so those are the kinds of issues that I think are you know, hard for the academy to solve, less so in, you know, maybe in industry.
I think the clinical space has had to solve this problem far more aggressively. Now, I'm not going to say efficiently in terms of fair, but in terms of privacy and all of those critical parameters, and with an enormous amount of legal pressure of, you know, malpractice suits and everything else. So they've had a lot. The challenge we found, so when we were building everything, you know, that was something we looked at a little bit, is what they had solved. Part of the problem that we encountered was that their solutions are very rigorous. They're very rigid, ossified. And so they work really well for like, you're doing the same thing again and again and again and again and again. We needed a solution that was incredibly dynamic, so that an experimenter could do something once that nobody had ever done before, and there weren't agreed on metadata and everything else, and we could ingest it into the system immediately without needing 6 months of committee meetings. So, but that's a group, you know, there are definite lessons there to be drawn from.
Yeah, so slight change. I'm in a genomics core at Dana-Farber. We get lots of, I need my data tomorrow, and then we post that data for them tomorrow, but they don't collect it for 6 months. Do you have any strategies or, yeah?
Yeah. Mine is we need the data tomorrow, and then 6 months later they say, oh, there was an error in my sample sheet. Can you re-demultiplex it? It's all wrong. So, yeah, I mean, so wherever we move the data for, like the links that we provide people are persistent. And so even if I re-demultiplex it, that same link will be valid.
So for us, if we're transferring externally, there's a cost associated with that, right? I don't know what Globus, we use DNA Nexus. As long as that is up in the cloud, we're paying for it to be there. So I can't just leave it there for 6 months.
So we use Globus to transfer, because Globus, you know, the data never leaves our on-prem storage. And so we are paying for it in a way, but we're not paying the cloud storage costs. If people ask me to move data to the cloud, I only accept their bucket. I never put it in my bucket, because yeah, I don't want to pay for it.
So I want to jump in because we have the same thing. And the way our sequencing core has solved this is they say, we have 3 weeks of storage. And if you don't get it in 3 weeks, then you're going to have to pay for us to re-demultiplex it off the VCLs.
Do you actually charge?
Yep.
So that's something, you know, Stuart's right down the street from me. That's the competition we have, and it's something we're worried about, that if we start charging for that, they're going to go to Stuart and not come back to me.
You charge a lot less than I do. We're already sending stuff your way.
Yeah. Thank you.
It's probably.
Hi. Looks like most of the data we are talking about, it's in-house data generated in the core, right? But when we talk about the data management, it could be any PI at university, and they may not be using the in-house core for generating the data. So, and I lead the bioinformatics core at the University of Virginia. More than 50% of the data I analyze, they are generated outside the university. So what is your take on that? How we support data management, that data is not generated at the university? Thank you.
So any project can have incoming and outgoing data, right? And so if it's, there's the easy case to the hard case. I think for the easiest case, it's in the public domain. It has an open source license from Creative Commons, a well-recognized means, easy. At the hard case, it's sort of, it has to be governed by a DUA with very restrictive proprietary terms and conditions. So we rely on the contract offices in that case to have an agreement. That can take a lot of time. You don't want to do that if you don't have to. So the more that we can say that something is de-identified, public domain, very frictionless. And then, you know, it goes up and up in terms of the regulatory burden. But I would consult your contract offices to facilitate if it reaches some of those more regulated proprietary use cases when it's incoming data. But from a risk standpoint, incoming data is a lot easier than outgoing data. So for us, we have clinical data sets, and we have a secondary data use committee that examines whether data collected for clinical purposes can be released and de-identified for external purposes.
Great. Well, and that brings up all of the different compliance regimes that you must need to, yeah, which is a good time to end this session, I think. Thank you. So thank you very much for attending. I really appreciate your engagement, and let's thank our panelists one more time.
Thank you.
Thank you. That's great. That was the audience. They were super.
Oh, that's how we fund our